Welcome! Today we will delve into integrin alpha 2, or ITGA2, a key cellular receptor that recognizes collagen. We will explore its expression, structure, signaling pathways, its crucial roles in health and disease, particularly in cancer progression, and the tumor microenvironment, and examine its potential as a therapeutic target. ITGA2, also known as CD49B, is a central player in how cells interact with their surroundings. It acts as a primary sensor for collagen in the extracellular matrix. When ITGA2 functions abnormally, it becomes a powerful driver of cancer growth, helps tumors hide from the immune system, and contributes to other diseases, making it an attractive target for drugs across multiple cancer types. ITGA2 is not limited to one cell type. It is broadly found in epithelial cells lining our organs, in endothelial cells that form blood vessels, and in mesenchymal stromal cells, which are involved in tissue repair. This widespread expression underscores its fundamental role in maintaining the proper architecture and balance of our tissues. At the molecular level, ITGA2 is a large protein that spans the cell membrane. Its business end is the extracellular Mabnus domain, which, with the help of magnesium ions, grips onto specific sequences in collagen. To work effectively, ITGA2 pairs up with another protein called integrin beta 1, forming a complete receptor complex dedicated to collagen binding. When ITGA2 binds to collagen, it's like turning a key. The receptor changes shape, sending a signal inside the cell. This recruits messenger proteins like FAC and SIRCC, which then activate major signaling highways such as the 1 pathway. This cascade ultimately instructs the cell's nucleus to turn on genes that promote growth and survival, a process that, when hijacked, fuels cancer development. ITGA2's influence extends beyond the cancer cell itself, into the entire tumor neighborhood or microenvironment. Here, it wears many hats. On immune cells like regulatory T cells, it's associated with suppression. It can hinder cancer-fighting CD8+ T cells. On cancer-associated fibroblasts, ITGA2 engagement triggers a cascade that activates these cells. Furthermore, ITGA2 signaling can ramp up the immune checkpoint protein PDL1 on tumor cells, effectively putting up a do not attack sign. Colorectal cancer provides a clear example of ITGA2's dark side. Elevated ITGA2 acts as a biomarker and a functional engine for metastasis. It allows cancer cells to grip more tightly to the surrounding matrix, facilitating their spread. Alarmingly, these cells can secrete a factor called TGF-alpha that further boosts their own ITGA2 levels, creating a vicious cycle that drives aggressive disease progression. Given its importance, blocking ITGA2 has been pursued therapeutically. Vedolizumab was one such attempt, an antibody designed to lock onto ITGA2. It was tested in various inflammatory and autoimmune conditions. However, despite the sound biological rationale, the clinical trial results did not meet expectations, leading to its discontinuation. This underscores the complexity of translating a good target into an effective drug, though it does not negate ITGA2's fundamental role in disease. More on targets sharing. See you next time.